All right, so today's video is going to focus on muscles and how they contract. There are a set of diagrams that are attached to this assignment as well in Google Classroom that could be beneficial to you. You may need to split your screen, and maybe have them open on one side of the screen. If you have access to a printer, you may want to print them and use them to help you take some notes. Okay, so we're going to start here with the motor unit. So the motor unit is just a group of muscle fibers under the control of one motor neuron. So basically it's one motor neuron. You can see, like, for instance, down here I've got motor unit B versus like motor unit A. Okay. So it's one motor neuron and all of the muscle fibers that that one motor neuron controls. So you can see, for instance, motor unit B, the purpley type one, is going to this one, and this muscle fiber, oh, can't see the red very well, and this muscle fiber here. And so that would be the motor unit. The neuron, which is the nerve that's under your voluntary control, so the motor neuron, and then all of the muscle fibers that it's going to innervate. When you contract your muscle, you're not contracting every muscle fiber all at one time, unless you're trying to exert maximum force. And so this is one of the ways that we can kind of help control the amount of force we put out and effort we put out, is that we aren't, it doesn't necessarily have one neuron that controls the entire muscle. So that way, if we need more strength, we can try to recruit more. If we just need a little bit, like say to pick up a pencil, then we only need to recruit a few fibers. And so this motor unit can help give us some of that control. All right, so we're going to look pretty closely here at what we call the synapse. Okay, and so that's how you spell synapse there. So the synapse is going to be basically where the neuron comes in contact or, signals another cell. And you'll notice in the image there, they don't fully come together. They aren't, right on top of one another. There's a little gap between them, and we're going to talk about all of those different parts of that as we work our way through this diagram. Again, this is a picture that you guys have in the diagrams that are associated with this assignment on Google Classroom, and it could be to your benefit to have these diagrams open and to be, trying to label and work with them at the same time. So I'm really going to focus on this diagram here. We'll hit the information that's listed up here, but I'm going to hit it as I just work my way through the diagram. So one of the things that, so as we look at the diagram, the yellow part here, okay, so this kind of yellowy looking part here, that's the neuron. That's the axon, which is the end of the neuron, coming down to the muscle. And so this, where it says sarcolemma right here, okay, this red part down at the bottom here, that's the muscle. And so this particular synapse is called the neuromuscular junction, because it's where a neuron and a muscle are coming together. And so at this neuromuscular junction, we've got a couple of things going on. First of all, in the neuron, you're going to have these synaptic vesicles. Okay, so synaptic vesicles, that's how you spell those right there. And those synaptic vesicles are going to contain acetylcholine. Acetylcholine is right here. It's abbreviated in your diagram there as ACH. Okay, so acetylcholine is the neurotransmitter. So acetylcholine is the molecule that transmits the signal from the neuron to the muscle cell. So the neuron transmits information electrically, but we wouldn't want the neuron to electrically transmit the information to the muscle cell. Basically, what it would be doing is shocking the muscle cell, and that would be really uncomfortable. So when the message hits the end of the neuron, these red arrows here indicate the signal coming down. When the message hits the end of the neuron, the synaptic vesicles here that'll contain acetylcholine, that'll trigger their release. And if you look in what's called the synaptic cleft, so the synaptic cleft is the space here between the neuron and the muscle cell. So if you look at the synaptic cleft here, you can see the acetylcholine molecules that have been released floating in that synaptic cleft. So you can see them, all those little green dots there, floating around in that synaptic cleft. So the signal came down and it triggered the release of the acetylcholine from those synaptic vesicles. And that acetylcholine is going to travel across the synaptic cleft, and it's going to bind to the receptors that are on the sarcolemma. The sarcolemma is just a fancy term for the muscle cell membrane. So the sarcolemma contains these receptors here for the acetylcholine. And you can see the purple receptors there. And when the acetylcholine binds to them, they open up. And so when that acetylcholine binds to those receptors open up and they allow, those are sodium channels, they allow sodium to rush in. So the acetylcholine binds to receptors on sarcolemma. The result then is that sodium rushes into the muscle. And so what that does is that causes this depolarization. See up here in your information here where it says depolarization takes place on the muscle fiber. So that depolarization is a result of that sodium rushing in. Okay, so basically sodium is an ion, right? And sodium is going to be is a positive ion, right? Sodium looks like that. So sodium is an ion. And what will happen is as that sodium rushes in, that sodium shifts the electrical charge of the membrane. So, for instance, if I have this membrane here, okay, and all this positive ions rush into the membrane, cross and move to this other side, then that's going to change the electrical charge of the membrane. And so what that is called is that's called our depolarization. And we'll talk a little bit more in detail with that when we talk about neurons and stuff. And we'll look at some graphs with this. But basically, let's just kind of introduce the term here. So this depolarization is going to change the charge of the membrane. And that's what's going to signal the muscle that it's time to contract. So all this acetylcholine rushing into all the sodium rushing in because the acetylcholine bound to the receptors there causes the depolarization, and that again triggers the muscle contraction to begin. Right? That's how the muscle gets the signal that, oh, I'm supposed to contract. Right? Is by that depolarization, that change in the charge of the membrane. Okay, so let's look a little bit closer at the actual muscle cell membrane. Okay, so we mentioned on the previous slide that the muscle cell membrane is called the sarcolemma. Okay, and so the sarcolemma is just a normal cell membrane, but it's on a muscle, so it gets this name, right? This sarcolemma. And you remember your muscle cells are striated. They have the dark and the light stripes. And we're going to look at kind of exactly what causes this. Again, these are also diagrams that you have that you may want to label, as we go through. You can see the multiple nuclei throughout this little portion of the muscle cell here. So now we're going to zoom in just a touch. So now I've zoomed in on that little tiny myofibril that was there. And so what I have here is I have the dark and the light bands that cause the striations. So the I band, you can see the I band right here. So the I band, okay, the I band is also called the light bands. So the I bands are the light bands. And the A bands in the middle there, those A bands are called the dark bands. So the A bands contain actin and myosin. Let me spell those for you. Right. Oh, they're spelled right there for you. So the A bands contain actin and myosin, where the I bands contain actin only. And so what we have on this myofibril here is we have a whole bunch of what are called sarcomeres. Okay, so there's your spelling there, sarcomere. So your sarcomere is your functional unit. This is what's actually contracting on the muscle cell. And so the sarcomere is going to run from Z disc to Z disc. So from here to here, that's a sarcomere, from Z disc to Z disc. Now at the Z disc, that's where the actin is anchored in. So Z disc anchors the actin. So the Z disc is where the actin is anchored in. The myosin is this dark red right here, and we're going to zoom in further on this in just a little bit. Okay. And then the H zone, which is in the middle, which is also sometimes called the bare zone, B-A-R-E. Okay, the H zone or the bare zone with that M line, this is an M line running right down the middle, okay, is where our myosin is going to be anchored in. And what happens is when our muscle contracts, these two filaments, this myosin and actin, they're going to just slide over one another, which is what's going to cause the shortening of the muscle and that contraction. So the myosin is what we call the thick filament. You can see this over here. Right, the myosin is our thick filament and the actin is our thin filament. And we're going to zoom in and look at this even closer. So we've zoomed in farther. So again, remember our sarcomere goes from Z disc to Z disc. The actin you can see is the yellow, the kind of, I'm sorry, the actin is the blue, looks a little bit like a double helix, like DNA. Right, so the actin is anchored into that Z disc. You can see that here. Okay, and then the myosin is in the middle. There's your M line, and then this area right here is the H zone or the bare zone. And so from here to here, oh, there we go. From there to there would be an A band, right, containing both actin and myosin. And then crossing the Z disc there would be an I band containing actin only. And that's where we get those striations from on the muscle cell. Okay, so zooming in again, even closer, right, here's the myosin or this thick filament, and you'll see it has these heads here, which are going to be important. We're going to look closer at how that's going to actually make them be able to slide across one another. There's that thinner actin filament, and you'll see there's a space here, right, that bare zone, which is going to be important because that's what actually allows it to contract, right? It allows the myosin to pull the actin towards the center so the actin has somewhere to go. Okay, so let's look at this a little bit closer. So this is what we call our sliding filament theory. And this sliding filament theory is that these two filaments, the actin and the myosin, will slide across one another, allowing for muscle contraction. And so if we look at picture A there, you'll see it says protein complex. So that protein complex is actually called the troponin tropomyosin complex. Okay, and how I usually describe this is that myosin and actin are like the annoying girlfriend and boyfriend that constantly have to be touching each other, right? So this myosin head right here loves the actin, and it wants to hold its hand all the time. And so the troponin tropomyosin complex prevents that. It blocks that and doesn't allow the myosin to hold the actin's hand, doesn't allow the two to bind together. So when the muscle cell receives that signal, okay, so remember we talked about the depolarization? Let's come over here. Okay. Sorry, I don't have my little pen, and so I'm trying to type so you can read it, but it slows the process down a little bit. Okay. Okay, so the troponin tropomyosin complex is covering up that binding site on the myosin. And so the first thing that needs to happen is that depolarization. Remember that depolarization came from when the acetylcholine, when that acetylcholine came in contact with those receptors, as the acetylcholine was released from the nerve and came across the synaptic cleft and bound to the receptors and opened up the channels. That caused, when all that sodium rushed into the muscle cell, right? Sodium rushes into muscle cell. So when all that sodium rushes into the muscle cell, that triggers that depolarization. Well, that depolarization then triggers the release of calcium, which is stored in the sarcoplasmic reticulum. So the sarcoplasmic reticulum is, why is that doing that? There we go. Okay, so the sarcoplasmic reticulum is basically a modified endoplasmic reticulum. And so if you remember, that's a cell part. right? So in the muscle cell, the sarcoplasmic reticulum stores a bunch of calcium. And so that depolarization signal triggers the release of the calcium from that sarcoplasmic reticulum. And you see that happening here in this second diagram B. See all this calcium being released. And that calcium, you'll notice when it binds to the troponin-tropomyosin complex, it moves the troponin-tropomyosin complex out of the way. Okay, so let's come down here. All right. So calcium release binds to tropomyosin complex. And so what that does is it moves, I'm just going to call it the TTC for abbreviation purpose now. So that moves the troponin-tropomyosin complex out of the way so the myosin head can bind to the actin. And so now, when that myosin head binds to the actin, I have to get this smaller. Okay, so now when that myosin head binds to the actin, it allows the actin, or the myosin then, to contract or kind of pull. You'll notice the arrows right here are those myosin heads. You'll notice they were kind of leaning out like this, and then they end up moving in like this. So they were kind of leaning out, and then they end up like pulling in because they're going to pull this actin. You'll notice the gap right here, the gap in the middle here is smaller because they're pulling the actin towards the center there. And so as they pull the actin towards the center there, that pulls, remember the actin is anchored into those Z discs. So that pulls those Z discs closer together then. And so that will cause the muscle as a whole to shorten. Okay, and so that's called our sliding filament theory. And it requires ATP. It requires ATP to move the myosin heads, as well as for the myosin heads to release the actin and grab again and pull again. So this is why it requires energy for us to do our muscle contractions. Okay, but this is how our muscle is going to contract. Those myosin heads, when they get the opportunity, are going to grab the actin and they're going to pull it more towards the center. Okay, and our bodies have what is called an all or none law, meaning that the muscle fiber, if it receives a signal that is strong enough for it to contract, it's going to contract. We're not going to have a partial contraction. We're not going to have like a minor contraction. If the muscle signal is strong enough to cause a contraction, then all of those motor fibers are going to contract. Okay, and so we get this all or none law. Okay, we've got a couple things left here. I know that the depolarization thing may still be a little bit confusing. The depolarization, the sliding filament theory thing may still be a little bit confusing. Remember, you have a book. Okay, feel free to go read that chunk in your book. Also, if there's a specific question or a specific thing that you didn't understand, feel free to email or remind message. Okay, so our slow twitch versus fast twitch fibers. So slow twitch fibers, they are going to contract slower. Okay, so these are going to be better for endurance, where our fast twitch fibers, they are going to contract faster. And so these are our type 2A. Those are our type 2A are considered intermediates. I don't know. Apparently this thing and I are going to have a fight. So our 2A are considered intermediate, and our type 2B, these are contract the fastest, so they contract very rapidly. They are most common in sprint athletes, but they fatigue very quickly. So your really elite athletes, they may have, like, for instance, your elite marathon runners, they may have more of these. They may be physically built with more of these slow twitch fibers, where your elite sprinters may have more of these type 2B fibers that contract very rapidly, although they don't allow for a lot of endurance. All right, and the last thing that we need to talk about here is our strength, power, and endurance of our muscles. So, strength is basically just the force the muscle can produce. There's like the long definitions there given to you by your book, but that's what strength is. It's the amount of force a muscle can produce, the resistance that someone is able to move. Where power is going to be basically how the shortening velocity during contraction. So the amount of force it's producing at the speed that it's producing it. So force and speed combined gives us our power, and our endurance is the amount of tension or force that can be produced over a period of time. And so the longer, the more endurance we have, the more tension we can produce over time. So, this is kind of the basics of how your muscles work. All the other things we're going to be doing with muscles in this chapter are going to be basically on your own, looking at different muscle functions as well as different muscle locations.